Welcome to Cut the Crop, keeping you in the know about everything you grow. Researchers, growers and other interesting folk delivering insights and information on topics ranging from agronomy to profitability. Brought to you by the Foundation for Arable Research.
Hello and welcome. My name's Alison Stewart and I'm your host of this session of Cut the Crop. And today we have one of our regular guests, Jo Drummond, a senior cereal researcher from FAR. Welcome, Jo.
Thank you, Alison.
Yeah. So today, I guess you're going to be talking about the risk of relying on the same fungicides for control of Septoria tritici blight. So I guess let's just start off and talk about where do we sit at the moment with how effective the fungicides are for STB.
Right. So at the moment, our chemistry is still effective, although we are seeing some sensitivity shifts in some mode of action groups. That means we actually have a bit of a call to action this season so that we can use our chemistry wisely and continue to use it in the years to come.
So overseas, there are much worse situations.
Oh, absolutely. And we have the opportunity to learn from the mistakes that have happened in other parts of the world and implement some really good resistance management strategies to help prevent us get into that same conundrum where they're losing modes of action.
So let's talk about the amount of shift in sensitivity that you've been measuring over the last how many years?
I think it started in about 2014. So we've got quite a lot of data going back over a number of years. And to kind of paint the picture for you, what we're looking at is we're looking at the sensitivity of Septoria tritici. So that's the pathogen that causes Septoria tritici blotch. And we're looking at that against a number of active ingredients that we commonly use in our programs across a number of mode of action groups because our programs are all sort of built on triazoles. So there's group 3 demethylase inhibitors, and then we dial in extra chemistry as we need it. So we dial in the likes of our group 7s, which are our SDHIs, and then we have other chemistry like our multi-site fulpit, so that's a group M4, and things like fenpicoxamide, which is a group 21, which is some newer chemistry to the market. So, and then we have other groups of chemistry that we're not looking at in this instance, so the strobilurins, so group 11. So there's a lot of terminology in a lot of groups, and it is really helpful for us to know what groups are happening and in programs and where and when that can give us some guidance as to how to interpret some of these sensitivity shifts and what we might need to do on farm this season to make sure they work for us.
So let's just talk about what you mean by a sensitivity shift and what that means to the product performing in the field.
Absolutely. So what happens when we start to see sensitivity shifts is basically what we've got out there is a big pathogen soup. And that pathogen soup is really adaptable. And when we talk about sensitivity shifts, what that means is it requires more of the same active ingredient to get the same amount of knockdown and control. So basically what we see is over time, you need gradually more and more and more chemistry to do the same job. And eventually the chemistry doesn't work for us anymore. And it's a slow pathway, and it's a pathway that's very difficult to stop in its tracks. And the best that we can do is slow it down as much as possible.
Yeah. So what is the strategy that we recommend to be able to slow it down? It's all linked to mode of action groups.
Absolutely. And it's going to be ever so slightly different depending on what mode of action group that we're talking about. And we'll start with triazoles because those form the backbone of our fungicide program. So those are our group 3s. And worryingly, these are the groups where we've seen the greatest amount of sensitivity shift. And on farm, you'll have noticed that probably way back in 2014 when we looked at epoxyconazole and prothioconazole. So those are the sorts of products that we see in the likes of Opus and its generics, so things like Stella and those types of products, and Proline, so that's and its generics as well. So there are a number of products out there on the market, and they all have different trade names, but they have the same active ingredients. So way back in 2014, we picked up that that the sensitivity shift in epoxyconazole was happening perhaps a little bit quicker than prothioconazole, and what that meant for us in our programs is that we started to see a lot more Proline. In more recent years, we've seen the introduction of mefentrifluconazole. I had to practice that name, which you would know as one of the active ingredients in Revistar and Revolution. So that's come to the market in more recent years. They all have the same mode of action group. So what that means for us is that we need to mix and alternate. So we need to mix our mode of action groups, and we need to alternate both within and between seasons. So the temptation is when something works really well for us is to use it all the time. We can't do that with our triazoles anymore. So we really have to mix. So if you're using one triazole at your growth stage 32 application, use a different one at your growth stage 39 application, and a different one again at your growth stage 65, 59 to 65. It's really, really important that we do this, because some of the cross-resistant pathways means that we can drive ourselves there quicker. And we also know that some can be mixed together and not others. Traditionally, there has been a school of thought that mixing triazoles with different cross-resistance groups is a strategy that can be helpful in terms of resistance management, but we don't know how the individual mutations within that pathogen soup are working, so we can't rely on some of that old wisdom. So now we need to focus on mixing our mode of action groups and making sure that our mixing partners are also effective, and alternating as if our lives depend upon it, because the life of your fungicide certainly does.
So you talk about cross-resistance. I mean, just for any listeners out there, just explain what you mean by cross-resistance.
So it's a little bit like within a family. What happens is that some active ingredients within the same family are going to travel down exactly the same pathway. So if you get resistance in one, you'll get resistance in the other at the same time. Others have slightly different cross-resistant pathways, so you can use them in tandem, and they might develop at slightly different rates. And that's why we see stacked azoles often marketed together, because they have different cross-resistance pathways. But because we don't know how those mutations are going to be working on each individual active, we definitely can't rely on that as the only strategy we use. So alternating and multiple modes of action is really, really critical if we want to save this chemistry, because it is our backbone. Without it, we are left with very high-risk chemistry, which leads ourselves to the SDHI situation, which thankfully is stable for the moment.
How, if you're a grower, where do you find the information on the mode of action group when you buy your product?
So it's on your label. It's on your product label. And it's a really good idea to get that into your head. And I know you will have all started thinking about this with things like herbicides. It's also really important to think about that with fungicides as well, because there's not that many groups that we use. And often resistance, particularly in that triazole group, that group 3, is actually driven by the number of applications. So if we can limit the number of applications as much as possible, in addition to alternating and mixing, that's going to be a really big step in understanding how resistance management works and making sure that we've got longevity in our chemistries. And if you're not sure, ask your rep.
Yes. Or come to a FAR.
Or come to a FAR field day, because we talk about this all the time.
I mean, so you're really talking about good stewardship, aren't you, of your products?
Absolutely. And we want these products to be working for us as long as we possibly can. And that might mean really honing in on which applications are going to be important to us and which applications are going to add value to our system, because there's actually an inherent cost in implementing resistance management strategies. That might mean the cheap application you thought you might be doing very early or very late in your season may no longer be economically sensible for you and your business.
Yes. So, it is reasonably complicated.
It's very complicated.
So, I mean, what are the simple messages and guidelines that you can give to growers?
Right. So the first guideline, we'll talk about triazoles first. Alternate within and between seasons. So when you're building your program, think of your most important leaf first and then work your way around it. Mix your mode of action groups and make sure that your mixtures involve multiple modes of action. Alternate within your season and between your season, and make sure with a DMI that you're using the appropriate rate to do the appropriate job. For an SDHI, again, it's very similar messaging. It's if you don't need to use 2 SDHIs, then use 1. But if you do need to use 2 SDHIs within a single season, make sure you choose different ones, and that messaging goes through from one season to the next. And I'm sure we've all got our favourites that work for us really well on farm, but it's really important to recognise that we do need to alternate our chemistries, and that's really, really important. And we also need to think about dialing in some of those other groups that we've talked about, like our Phoenix and our Fempicoxamid, because that helps reduce selection pressure.
So if a grower wants to find out more about this, where's a good place to find information on how to manage fungicide resistance in cereal crops?
Well, we're currently working on updating all our strategies based on the information we've received this season. So keep a watchful eye out on our FAR Weekly and our Crop Action newsletters, because those will be updated throughout the season. And come along to our field days. So we'll be having events through September and October where we'll dig into this a little bit deeper to hopefully give you some guidance on farm and actually walk a crop and actually look at what pressures we have in front of us and talk about how we can manage our system based on all of the different factors that we need to think about when we're planning fungicide programs, because actually chemistry is the last thing we think about.
Because if we don't do this, then we will end up in a situation similar to, for example, some of the cereal growers in the UK, where they have almost nothing left available to them that actually is performing well.
And that's a position that we really want to avoid. And one of the things I sometimes think about, and I get growers saying this all the time, is, but I want that extra 100 kilos of yield. If we break these chemistries, we're not talking about 100 kilos of yield. We're talking about 2 tons or 3 tons or 4 tons. So it is in our best interest to preserve this chemistry as much as we can, because I don't want to have to stand up in front of growers and say that we're out of options. It's really important for us to take the learnings from other parts of the world while we still have a chance, because we do have a chance, but we do have to fix it, and we do have to fix it pretty soon.
Yes. And that is one of the, I guess, the rationale of why we're also looking at some of the biological products.
Absolutely. So we have screening trials in place at the moment and replicated trials as part of the Lighter Touch program, where we're trying to identify options that we might be able to dial in at timings that relieves the pressure on that chemistry to try and help us build programs. Because depending on what your pathogen pressure is, you may be able to dial in a biological to help get you over the line.
Yeah. And it's interesting because, sometimes we do get challenged by our growers about, why are we bothering with biologicals? The chemicals, are working really well. They're the mainstay of the Cereal Crop Protection Foundation. And the reason we're doing it is because we want to keep and protect the chemicals.
Absolutely. No, it's not a one or t'other. It's actually having a program that's built on sound resistance management, so you can keep your big guns for when you need your big guns. Yeah. That's really what it boils down to. Keep the important things for the important parts of your program. And it's about controlling what you can control and limiting the amount of risk in other parts of your season and other parts of your program as much as possible. So we're trying to give you as many options as we can so that you can keep growing, because that's what we want.
Yes. So any little final sort of comments that you'd like to say, summarising key messages?
Key messages: mix your modes of action, alternate within and between seasons, consider your timings and the importance of those timings and the importance of those leaves. Think about your cultivar, think about your weather, and whether or not you need to go on with an application, because everything that we can do to slow down resistance in our key mode of action groups is going to be money well spent.
Absolutely. Thank you very much, Jo.
Thank you, Alison.
Yeah, and thank you, listeners. Have a great day on the farm.
Thanks for tuning in to Cut the Crop, presented to you by FAR. If you'd like to know more about any of the topics discussed or have any suggestions for future topics, go to www.cutthecrop.co.nz.